Imagine, if you will, a scenario that pushes the absolute limits of survival. You are starving. The food supply hasn't just dwindled. I mean, it has completely vanished.
Right, like a total catastrophic famine.
Exactly. And you are surrounded by thousands of your neighbors in this completely desolate environment. Every single one of you is facing imminent starvation. But there is a way out.
Okay, what's the catch?
Well, your only chance of survival is to team up with all of those neighbors and physically build an escape tower, to get up and out of the wasteland, to reach an environment where food might still exist.
It sounds like a pretty classic cooperative survival problem, honestly. But the mechanics of this particular tower, well, they change the moral calculus entirely.
They really do, because there is a massive, horrifying catch to this escape plan. In order for the tower to stand, 25% of the population has to volunteer to die.
Wow.
Right. A quarter of you must literally sacrifice your own bodies, essentially cementing yourselves together to form the rigid, dead structure of the tower's base and stalk.
Just so the others can survive.
Exactly. You do this so the remaining 75% can climb up your dead bodies, escape, and live. So put yourself in those shoes, Would you do it? Would you volunteer to be part of the dead structural foundation?
I mean, from a purely self-interested standpoint, the answer is usually no.
Right. And this is where the psychology and really the biology kicks in. What exactly stops someone in that crowd from pretending to help, acting like they are going to sacrifice themselves, but then sneaking out the top of the tower at the absolute last second?
Yeah, if you can reap all the benefits of the escape without paying the ultimate price, the temptation to cheat is just overwhelming.
It's the ultimate freeloader problem.
It is the ultimate evolutionary dilemma. The temptation to let someone else pay the price while you survive is mathematically profound. I mean, if a cheater survives and the altruist dies, the cheater is the one who passes on their genes.
So eventually, the cheaters just take over.
Exactly. Over time, the cheaters should inherit the earth, and cooperation should just completely collapse.
And that dark temptation, that inherent vulnerability in any cooperative system, is the starting point for our mission today. So, welcome to today's deep dive. We are setting out to explore one of the greatest, most enduring mysteries in all of evolutionary biology.
Which is how complex cooperative societies actually manage to survive the constant, inevitable threat of cheaters and freeloaders.
Exactly. We are diving into a really fascinating stack of biological research today. We'll be looking at a foundational 1982 paper by Leo Buss on somatic cell parasitism, getting some incredible insights from John Maynard Smith and Eörs Szathmáry's 1997 book, The Major Transitions in Evolution, and taking it all the way to a pivotal 2007 field study by Gilbert and colleagues.
The stakes here are incredibly high, too. We aren't just talking about, behavioral quirks or abstract social theories. We're looking at the exact microscopic moment where life makes the leap from being single, selfish cells into complex, cooperative, multicellular organisms.
Because, you, me, the dog sleeping on the rug, multicellular life is just a massive cooperative society of trillions of cells.
Exactly. It's a miracle it works at all.
So to understand how that transition doesn't just instantly collapse under the weight of biological selfishness, we need to look at an organism that is currently straddling that exact evolutionary boundary.
We're going to look at the bizarre, microscopic, and surprisingly dramatic world of slime mold.
Yeah, and they're such a wild thing to study. I think we need to start by defining what these organisms actually are. Because when I hear slime mold, I basically picture something gross and static growing under a damp log.
Right, but the biology of these organisms is so much more dynamic than that.
So we are specifically looking at eukaryotic slime molds, like one called Dictyostelium, and comparing them to prokaryotic myxobacteria. But before we even get into their behavior, we should probably clarify those terms.
Yeah, it's a crucial distinction. Let's break it down. Prokaryotic cells, like bacteria and myxobacteria, are the older, simpler form of life. They don't have a nucleus. Their DNA is just, free-floating inside the cell.
Like a big open room.
Right. You can think of a prokaryotic cell like a studio apartment. Everything happens in one big open space.
Okay, so eukaryotic cells, like the Dictyostelium slime mold, are more complex.
Much more. Eukaryotic cells have their DNA safely locked inside a nucleus, and they have various specialized compartments or organelles doing different jobs. If a prokaryote is a studio apartment, a eukaryote is a sprawling mansion with a kitchen, a library, and specialized rooms.
And eukaryotes are what plants, animals, and fungi are made of, right?
Yeah, exactly.
Okay, so we are looking at two entirely different branches of the tree of life, the studio apartments and the mansions. But what's fascinating here is that despite being fundamentally different types of cells, they have independently evolved strikingly similar behaviors in the wild.
Yeah, convergent evolution. It's amazing.
Both of them live in wet soil. Neither of them can swim because they lack flagella or tails, so they have to slowly crawl or glide across solid surfaces. And their normal day-to-day life is basically just crawling around as solitary single cells, eating other microbes.
Right. They were just microscopic learners, completely autonomous. For the vast majority of their lives, they act entirely in their own self-interest. They hunt, they eat, they divide in two. That is the entirety of their existence.
But the trigger that completely upends their solitary existence is starvation. When the local bacteria are depleted, everything changes.
Specifically, the research points to amino acid starvation.
Okay, let's unpack this. Amino acids are the building blocks of proteins, right? So when the environment runs out of them, these cells hit a biological panic button.
They absolutely do. They stop reproducing entirely. Their normal cellular division halts, and they initiate this spectacular developmental phase.
And this is where the mechanics become truly incredible to me. Because they are solitary and dispersed all across the soil, they have to somehow communicate with thousands of other independent cells to coordinate a massive physical aggregation.
Yeah, how do you organize 1 million blind amoebae?
Right. How does a brainless, solitary amoeba shout, "The food is gone, gather round"? Well, in Dictyostelium, they do this by releasing a chemical molecule called CAM-MP, or cyclic adenosine monophosphate, into their environment.
But it isn't just a steady, continuous leak of a chemical.
No, because if it was a steady leak, the chemical would just slowly diffuse outward, right? It would just be a gradient.
Yeah, and it would be very hard for distant cells to figure out exactly where the center was. Instead, they release it in pulses.
It's an oscillatory process. It creates these visible traveling waves of chemical signals moving through the soil. I like to think of it like a giant microscopic stadium wave, where starvation is the music stopping in a game of musical chairs. It forces everyone to suddenly rush together.
That's a great way to picture it.
I really want to break down exactly how this mechanism works because it's just brilliant. Imagine you are standing on a mountain peak at night, and you need to signal thousands of people scattered across the surrounding hills to come to your exact location.
So you light a massive flare.
Right. You light a flare. The people in the nearest hills see your flare. They immediately light their own flares to pass the signal on, and then they start walking toward your original light.
But there is a mechanical problem with that analogy if we don't introduce a refractory period.
Exactly. If everyone just keeps their flares lit, the whole mountain range is glowing and no one knows which direction the center is anymore.
Right.
It's just a giant blob of light.
Right. It would just be chaos. So the amoebae have a built-in biological timer. After an amoeba detects the CAM-P chemical and releases its own pulse, it becomes temporarily blind to the chemical for a few minutes.
It literally turns its receptor off.
Yes. That refractory period is the genius of the entire system. It ensures that the chemical wave only travels outward, away from the center, while the amoebae themselves only crawl inward toward the center.
So the cell feels the chemical hit its front, it fires its own pulse to alert the cells behind it, and then it marches forward while ignoring any signals coming from behind.
It's like a chain reaction of flares, where once you light yours, you put on blinders so you can only see the flare in front of you.
That is wild. This intricate mechanism allows hundreds of thousands of dispersed solitary amoebae to suddenly turn and rush together toward a single central point without ever actually touching each other initially.
The chemical gradient does all the heavy lifting for the synchronization, and then once they finally reach that center point and aggregate, they physically adhere to one another.
They become a solid thing.
Yeah, they form this cohesive, migrating mass of cells that biologists actually call a slug.
And it literally looks like a tiny translucent slug, right? It crawls along the substrate as a single unit.
It totally does. It behaves like a single organism. It has a front and a back. It crawls toward light and heat, seeking the optimal surface, usually a patch of soil exposed to the air, or maybe a piece of dung.
And once it finds the perfect spot, it stops crawling and initiates culmination. It stands upright and transforms into a mature fruiting body.
We are talking about a structure maybe 3 millimeters high, which to us is nothing. You'd barely see it.
But to an amoeba, That is a skyscraper. I mean, it is made up of anywhere from 100,000 to 1,000,000 individual cells.
And this is where the economic concept of the division of labor gets applied to microbiology. To build this towering, fruiting body, the cells must differentiate. They can no longer all be identical. They have to take on specialized jobs.
Which brings us back to our dystopian opening scenario. In the myxobacteria, some of the cells literally lyse. They break open their own cell walls and die, spilling their rigid contents to help form the structure.
Right, extreme sacrifice.
And in the eukaryotic slime mold Dictyostelium, it's highly orchestrated. About 25% of the cells sacrifice themselves. They crawl to the bottom and the center of the mass, and they form a rigid cellulose-lined stalk.
They essentially build a plant stem out of their own bodies, give up any chance of ever reproducing or passing on their genes, and they just die.
They become nothing more than dead structural support. While the remaining 75% of the cells climb up the outside of that stalk to the very top.
And there, they transform into spores. Now, these are haploid, asexual spores.
Let's clarify haploid for a moment for the listeners.
Sure. Human cells are diploid, meaning we have two sets of chromosomes, one from our mother, one from our father. Our sperm and egg cells are haploid, meaning they only carry one set.
Okay, got it.
So these slime mold spores are haploid. They contain just a single set of genetic instructions. They sit in a little sticky droplet at the top of the stalk, completely dormant, just waiting.
Waiting for what, exactly?
Waiting to stick to the leg or the carapace of a passing insect, like a fly or a beetle. That insect will eventually fly off and land in a new, nutrient-rich environment, brushing the spores off.
And then the spores germinate back into solitary amoebae and the whole cycle starts over.
Exactly.
So we really have to ask why this evolved. Because the sacrifice is immense. A 25% mortality rate just to build a stalk is a massive evolutionary cost. What is the payoff?
It depends on which organism we're looking at. For the prokaryotic myxobacteria, the aggregation is largely about cooperative feeding. We mentioned earlier they eat other microbes, but they don't have a rigid internal skeleton, so they can't just engulf their prey like a Pac-Man.
Oh, right. That's called phagocytosis.
Exactly. They can't do that.
So how do they eat then?
They secrete digestive enzymes into their environment to break down the prey externally, and then they absorb the nutrient soup. But if a single myxobacterium secretes enzymes, those enzymes just float away in the moisture of the soil. It's incredibly inefficient.
So they need numbers.
Right. By clustering together in massive numbers, they pool those enzymes. They essentially create a highly concentrated, shared digestive space. It allows them to dissolve large colonies of other bacteria that they could never tackle alone.
Wow, they hunt like a microscopic wolf pack.
Precisely. But for the eukaryotic slime molds like Dictyostelium, this elaborate sacrificial tower building is almost entirely about efficient spore dispersal.
Just getting off the ground.
Yeah. A single amoeba sitting flat on the dirt has almost zero chance of being picked up by a passing fly. Getting just 3 millimeters off of the ground, getting above that microscopic boundary layer of stagnant air and debris on the soil surface exponentially increases their chances of hitching a ride to a better environment.
Okay, so the mechanism is brilliant and the evolutionary advantage is clear. But this entire life cycle presents a staggering fundamental vulnerability.
It really does. It's a very fragile system.
I want to contrast this with a human being. You and I grew from a single fertilized egg. That one cell divided into 2, then 4, then 8. Because we grew from a single point of origin, every single cell in our body, our heart cells, our skin cells, our brain cells, is essentially a genetic clone of the original.
Right. They all share the exact same DNA. They are a team from day one.
And because they share the exact same DNA, their evolutionary interests are perfectly aligned. Your heart cell doesn't, want to rebel against your brain cell because helping the brain survive helps the body reproduce, which passes on the heart cell's DNA too.
It's all the same genetic vehicle.
Right. But the slime mold doesn't grow from a single egg. It is formed anew in every single life cycle by aggregation. Millions of independent, free-roaming cells in the dirt who have just spent the last few weeks living entirely selfish lives are just walking up to each other and agreeing to form a single body.
And that is the crux of the issue. Because the organism forms by aggregation, it openly invites different genetic lineages to mix together.
It's just whoever happens to be in the neighborhood.
Exactly. If 2 different strains of amoebae happen to be living in the exact same millimeter of soil when the food runs out, the chemical wave will draw them both in. They will aggregate together.
And the moment you mix different genetic lineages, you open the door to evolutionary conflict.
You open the door to cheating. If strain A and strain B are in the same tower, and strain A can somehow trick strain B into forming the dead stalk, strain A gets all its cells into the spores and basically wins the evolutionary lottery.
Which brings us to the core mystery of this deep dive. What happens when a mutant cell enters the mix and simply refuses to cooperate? Like, how devastating is it?
It can be catastrophic.
Let's look at Leo Buss's incredible 1982 paper. He was studying natural populations of a species called Dictyostelium mucoroides. And he didn't just model this on a computer, right? He actually went out and collected 14 bundles of soil cores from the wild.
He had his hands dirty. And in one of those natural soil cores, he found a mutant that was structurally bizarre.
It was a completely stalkless variant. Instead of building that beautiful 3-millimeter tower with a rigid stalk, when this mutant strain starved, it just produced a sad little ball of spores lying completely flat on the dirt.
It had lost the genetic instruction to sacrifice cells for a stalk.
And functionally, that is a dead end. Without a stalk to lift the spore mass into the air, those sticky spores have absolutely no way to adhere to the insects we talked about earlier.
On its own, this mutant cannot disperse. If it were completely isolated in a patch of dirt, it would eat the bacteria, starve, fail to build a tower, and that entire genetic line would simply go extinct right there.
But Leo Buss forced an interaction. He set up an experiment in the lab to test what he called cell lineage competition. He took this pathetic, flat, stalkless mutant and mixed it in a petri dish with a normal, healthy, tower-building strain of the slime mold.
He created what biologists call a chimera.
Just to define that for everyone, in biology, a chimera is a single organism made up of cells with more than one distinct genotype, right?
Right. It's named after the mythological beast that was part lion, part goat, part snake. Here, it's a single slime mold slug made of 2 completely different genetic strains of amoebae.
So Buss creates this chimera, forces them to starve, and waits to see what happens. And in the presence of the normal cells, the stalkless mutant didn't just survive, its frequency in the population rapidly increased.
Because it operates as a textbook somatic cell parasite?
Okay, how exactly does it pull off the heist? Because it seems like it's just doing nothing.
Well, when the mixed population starves and aggregates, the normal cells follow their evolutionary programming. They detect the signals, they assess their position, bravely march to the bottom to die and build the stalk.
Doing what they're supposed to do.
Right. But the mutant cells contribute 0 cells to the dead stalk. Instead, every single mutant cell just climbs up the stalk built by the normal victims and becomes a reproductive spore.
But if it takes over the gene pool, there are no normal cells left to build the stalk. And the mutant can't build a stalk on its own, so the whole species should just collapse.
It's a paradox.
Yeah, why hasn't this parasitic mutant accidentally wiped out the entire slime mold species by now?
That is the exact paradox Buss was grappling with. If the cheater becomes too common, it mathematically destroys the group's ability to make a functional structure.
How so?
There simply aren't enough normal cells left to hold up the sheer physical weight of all the parasitic spores. The fruiting body buckles.
It just falls over.
It collapses right into the dirt, dispersal fails entirely, and the entire population, normal cells and cheaters alike, crashes.
Wow. So the cheater is trapped by its own success.
But that brings up a crucial problem with lab experiments. Buss forced them together in a dish.
Right. It's totally artificial.
Exactly. If this cheater is so devastating in a dish, how do we know it actually behaves that way out in the mud?
And this is where we pivot to the wild, because John Maynard Smith, a legendary evolutionary biologist, proposed a theoretical solution to this exact problem. These amoebas are incredibly slow. They crawl at an agonizingly microscopic pace. Therefore, when you see a mature fruiting body in the wild, they likely descended from a single solitary ancestor that landed there a few days ago and just divided a bunch of times in that one specific localized spot.
Which means all the cells in that immediate vicinity would be highly related to each other. They would essentially be a family of clones. This is the concept of kin selection.
Let's explore how kin selection actually defeats a cheater, because I think it's brilliant.
Think about the mechanics of the mutation. Let's say a mutation arises in one cell that says, don't build the stalk, just become a spore. If that cell divides and its daughters divide and they form a localized colony of clones, every single cell in that colony has the cheater mutation.
So when they starve and try to aggregate, nobody builds the stalk.
Precisely. They all stubbornly wait for someone else to build the stalk. The structure fails and they all die. Cheating only works if you can exploit a genetically different neighbor who doesn't have the cheating gene and is willing to do the hard work.
Okay, let's put an analogy on this because it clarifies the mechanism perfectly. Here's where it gets really interesting. It's exactly like a high school group project.
Oh, everyone remembers those.
We all know how the group project dynamics work. If the teacher randomly mixes all the students in the entire class together into groups, which is the equivalent of a low relatedness, highly mixed genetic environment, the slackers and the cheaters get a massive benefit.
They do absolutely no work, they exploit the hard workers in their group, and they get the same A on the project.
The slackers thrive when they are mixed with strangers. But if the teacher knows exactly who the slackers are and intentionally forces all the slackers into one single group and all the hard workers into another group, which is the equivalent of a high relatedness clonal environment, the dynamics flip completely.
The slacker group absolutely fails the class because no one does the work.
And the hard worker group gets A's. Maynard Smith suggested that nature acts like that strict teacher. Because the amoebae move so slowly, nature naturally groups them only with their closest kin, meaning the cheaters only end up cheating themselves, and natural selection purges them from the population.
It perfectly explains how cooperation can be maintained. But science isn't satisfied with elegant theories.
No, we need proof.
Exactly. It requires empirical evidence. Does this actually hold up in the messy, chaotic reality of a forest floor?
And that question leads us directly to the pivotal 2007 paper by Gilbert and his colleagues at Rice University. They decided to take Maynard Smith's elegant theory out of the realm of speculation and actually test it in the dirt. And to do that, they had to embrace some truly unglamorous fieldwork.
Unglamorous is certainly one word for it. They went out to the Mountain Lake Biological Station in Virginia, which is this dense Appalachian forest environment. And they spent their days walking the forest floor, actively hunting for deer dung.
Specifically, white-tailed deer scat. But why deer dung? Why not just scoop up some soil?
It all comes down to bacterial density. A slime mold needs to eat a massive amount of bacteria to reach a population size large enough to form a fruiting body.
And soil doesn't have enough.
Normal forest soil has bacteria, but it's really spread out. A single pellet of fresh deer dung, however, is essentially a concentrated bacterial explosion. It is the absolute perfect, nutrient-dense feast for slime molds to thrive and reach the massive numbers required for their social cycle.
So they collect these dung pellets, carefully place them on sterile agar plates in plastic boxes, and incubate them in the laboratory, trying to simulate the humidity of the forest. And the visual details they recorded from this incubation are they're fascinating.
Yeah, it wasn't just slime molds on dung.
No, they didn't just find slime mold fruiting bodies growing on the dung itself. As the dung incubated, fly maggots that had been laid in the scat hatched and crawled around, eventually dying. The researchers documented slime mold fruiting bodies growing directly out of the decaying bodies of these dead flies and maggots.
It really paints a vivid picture of the opportunistic, cutthroat, microscopic ecosystem these organisms inhabit. They are feeding on the bacteria that are breaking down the dung and the dead insects.
But the core of their experiment wasn't just to watch them grow. It was genetic analysis. They collected 88 wild fruiting bodies directly from this dung. And their guiding question was, are these fruiting bodies mixed up chimeras full of strangers, like Bus made in his petri dish?
Or they're families of clones.
Right. To answer that, they had to genotype them. And this is where the methodology gets incredibly precise. They analyzed these fruiting bodies across 3 highly polymorphic microsatellite loci.
Okay, I actually need to pause you right there and define that, because polymorphic microsatellite loci is a lot of jargon.
Fair enough. Please do.
Think of a genome like a massive book of text. Most of the text, the genes that code for essential proteins, is identical from one amoeba to the next. But there are specific small regions of DNA that don't code for proteins. And these regions consist of short sequences of letters that just repeat over and over.
Like ATC, ATC, ATC.
Exactly. And those repetitions change from individual to individual. They are highly polymorphic, meaning they vary wildly in length across different lineages. One strain might have 5 repeats, while a strain from the next dung pellet over might have 20 repeats.
I see.
By looking at just a few of these specific locations, the loci scientists can create a unique genetic fingerprint for any individual. It's exactly how human paternity tests or forensic DNA profiling works.
Okay, so they're essentially swabbing the DNA of these microscopic fruiting bodies to run a paternity test on the colony.
Yes, exactly. And the results they got back were stunning. Out of the 88 fruiting bodies they tested, 77% showed single bands on the genetic test.
Meaning they were entirely clonal. Every cell in the entire tower was identical.
Yes, but they wanted to be absolutely rigorously sure. Testing a whole crushed-up fruiting body might mask a tiny minority of cheaters hiding inside.
Like a needle in a haystack.
Right. So they took another 75 fruiting bodies and painstakingly genotyped the individual microscopic spores from within them. Out of those, 92% consisted of a single pure clone.
The mathematics they derive from this data are what truly validates kin selection theory. Based on the whole fruiting body analysis, they calculated a minimum relatedness of 0.86. But when looking at the individual spores within all the tested bodies, the relatedness was a staggering 0.975.
That is a profound number in evolutionary biology, and we really need to unpack why. Relatedness is measured on a scale from 0 to 1. A relatedness of 1.0 means you are comparing two identical clones.
And a relatedness of 0 means you are comparing two completely unrelated strangers.
Exactly.
Let's put that in human terms to really ground it. My genetic relatedness to my own biological child is 0.5 because they get half their DNA from me and half from their other parent. My relatedness to a full sibling is, on average, 0.5. The only humans that have a relatedness of 1.0 are identical twins.
Right, so a relatedness of 0.975 means that out in the wild, when an amoeba crawls into an aggregate on a piece of deer dung to help build a tower, almost every single cell it bumps into is essentially its identical twin.
I really want to let that sink in. 0.975. That relatedness is exceptionally high. As you pointed out when we were looking over these sources earlier, that number is actually higher than the genetic relatedness found in most eusocial insect colonies, like highly cooperative beehives or ant mounds.
That extreme, near-perfect clonal population structure acts as an invisible, impenetrable shield against the stalkless mutant parasites.
Because, just like the high school group project, all the mutant cheaters are forced to group only with other mutant cheaters, and they fail to build a stock and die.
Exactly. They cheat themselves into extinction.
But this raises a really important scientific question. The field data from the dung clearly shows that relatedness is incredibly high in nature, but correlation isn't causation.
Well, to prove that, Gilbert's team had to play God in the laboratory. They couldn't just observe, they had to break the system on purpose.
They had to artificially lower the relatedness and introduce an assassin into the population to see what would happen.
And they found the absolute perfect assassin. They used a specific mutant strain known as fbxA-. This mutant has a very specific genetic knockout. It is entirely missing an F-box protein.
We should probably explain what an F-box protein actually does, because it illuminates exactly how cheating happens at a biochemical level. Inside every cell, there is essentially a molecular garbage disposal system called the ubiquitin proteasome system.
Right. When a cell needs to get rid of a protein that has done its job, it tags it for destruction.
It puts a little garbage tag on it.
Exactly. And F-box proteins are the molecules that act as the cellular garbage men. They are the ones that identify specific target proteins and slap the garbage tag on them so the cell knows to destroy them.
Okay, so this mutant is missing a specific garbage man. What was it supposed to be throwing away?
The F-box protein that is missing in this mutant is normally responsible for tagging and degrading a specific enzyme called a phosphodiesterase during the developmental phase.
And why is that important?
Because that phosphodiesterase enzyme controls the cell's internal clock for development. During the chaotic aggregation phase, as the cells are deciding who becomes stalk and who becomes spore, the degradation of that enzyme is the signal that tells the cell, okay, your time is up, you're at the bottom, it's time to sacrifice yourself and become a stalk cell.
Wow. So because the fbxA- mutant is missing the garbage man, the enzyme never gets degraded. The garbage piles up and the internal signal to sacrifice never goes off.
Exactly. The cell is biochemically blind to the sacrifice signal. It is the ultimate parasitic profile. It just keeps marching toward the top to become a spore.
And importantly, the researchers didn't just assume it was a cheater, they rigorously tested it to rule out other advantages. You might wonder, what if this mutant just grows and divides much faster than normal cells?
Right. If it just reproduces faster, it would naturally take over the population. And it wouldn't actually be cheating during the tower building phase.
But they controlled for that. They placed the mutant and the wild-type cells in a nutrient-rich liquid culture and tracked their vegetative growth rates. The wild-type and the mutant grew at the exact same speed. The mutant specifically and exclusively gains its advantage during the altruistic stalk-forming stage of development.
Furthermore, they prove that on its own, fbxA- is developmentally deficient. If you put it on a plate by itself without wild-type cells to exploit, the chemical confusion inside the cell prevents it from forming a proper structure, and it produces almost no viable spores.
It is an obligate parasite. It needs a victim.
So Gilbert's team set up the ultimate Thunderdome experiment in the lab. They took this fbxA- biochemical killer and mixed it with normal, cooperative, wild-type amoebae.
They forced the relatedness down to near zero.
And the results were an absolute biological bloodbath for cooperation. The researchers tracked what happened to the overall productivity, meaning the total number of spores produced by the group, as the frequency of the mutant increased.
And as fbxA- became more common, the total spore production just plummeted.
The fruiting bodies became poorly developed, they were misshapen, they collapsed under their own weight, or they completely failed to culminate into a tower at all.
It is a textbook, microscopic example of the tragedy of the commons. The cheater was winning the battle inside the group, stealing all the resources, but by doing so, it was systematically destroying the group's ability to survive in the process.
But because the researchers were carefully manipulating the frequencies in the lab, they were able to calculate the exact mathematical threshold for survival. They plotted the evolutionary fitness of the mutant on a graph against the genetic relatedness of the group.
And what did the math say?
They found that this deadly fbxA- cheater can only successfully invade and take over a population if the genetic relatedness of that population drops below 0.25.
Wow.
Yeah. If the relatedness is above 0.25, the kin selection effect is strong enough to purge it.
This is the crucial tying it all together finding. The mathematical threshold for the cheater to survive is 0.25. But we already know from their painstaking fieldwork swabbing DNA off the deer dung that the actual wild relatedness of these amoebae is over 0.86 and almost always around 0.975.
Therefore, the fbxA- mutant is mathematically locked out of nature. The natural population structure is so strictly clonal, so tightly knit, that a cheater like this can never gain a foothold.
But, whenever I read a lab study like this, where scientists are knocking out specific genes and mixing things in sterile petri dishes, I always have a lingering doubt.
What's that?
How do the researchers know for sure that this fbxA- mutant wasn't just secretly thriving out in the wild on the deer dung in some weird way they couldn't replicate in a sterile lab? Nature is incredibly complex. Maybe the mutant has a trick we haven't seen.
It is a vital question, and it's one the researchers fully anticipated and addressed. If you dig deep into the supplementary information of their paper, they detail a specific secondary experiment to test exactly this scenario. They didn't just test the mutant on sterile agar jelly.
What'd they do?
They took 5 million fbxA- mutant cells and placed them directly onto sterilized deer dung pellets to perfectly simulate their complex natural environment, just without the normal cells.
And the result?
Total abject failure. Out of all those millions of cells on all those dung pellets, only one single dung pellet produced any visible structures whatsoever. And when the researchers analyzed those structures, they contained absolutely zero viable spores.
So the mutant completely and utterly failed in the wild environment because it didn't have normal cooperative cells to exploit.
Exactly. Furthermore, to definitively, absolutely prove that these cheaters aren't somehow lurking undetected in the wild gene pool waiting for a chance to strike, they went back to their wild samples.
Oh, they checked the wild spores.
Yeah. They took over 3,300 wild spores collected from the forest floor, germinated them, and actively screened them looking for the fbxA- developmental defect.
And out of 3,316 wild spores pulled straight from nature,
they found absolute zero mutants. The shield holds. The relatedness barrier is completely impermeable in the wild.
It's an incredible defense mechanism. Against all odds, multicellular life is allowed to thrive.
It is a delicate, continuous, microscopic arms race, but it dictates the rules for all complex life on this planet.
And that leaves us with a final, slightly provocative thought to mull over, building on everything we've discussed from Buss, Maynard Smith, and Gilbert. Look at human civilization. We are a massive, hyper-complex, highly cooperative society built almost entirely on daily interactions with genetically unrelated strangers. Your relatedness to the person driving next to you on the highway is virtually zero.
You buy food grown by strangers. You drive on roads built by strangers. You trust your very life to strangers in hospitals and airplanes.
We defy the basic biological rule of cooperation every single day. So if the fundamental laws of biology dictate that low relatedness inevitably invites system-destroying cheaters. The freeloaders, the thieves, the parasites are human social structures, essentially just artificial defense mechanisms.
That's a fascinating question.
Our laws, our police forces, our complex moral codes, our religions, and our online reputational systems, are they essentially just artificial DIMs that we've had to desperately invent to prevent our own societal collapse? When you really think about it, are we just hacking the fundamental rules of evolution every single time we choose to trust a stranger?
It's a profound way to look at it. It reframes the entirety of human culture and law as a desperate, magnificent biological workaround to a microscopic problem.
It really does. Thank you all for joining us on this incredibly deep dive into the hidden rules of cooperation, from deer dung to human civilization. Keep exploring, keep questioning the hidden biological systems operating all around you, and we will see you next time.